my screen shortly to actually introduce to this short webinar here on the Working Party on Quality by Design. We will have 3 talks talking here, and I try to find the umbrella. We will have quality by design tools for the optimization of biopharma processes using system modeling, machine learning, and digital twins here. I think that's the umbrella, methodological umbrella, which we have here. And of course, for all of you which are probably not so much familiar with quality by design, I just give you a very short introduction to that. Always with my cycle, I go along this line, and quality by design is, in my opinion, to keep that very short, to have process understanding here between, let's say, the process variables on the one hand here on the quality attributes, KPIs probably, and its respective process parameters, which are probably here, or raw material attributes. We have to find that link, and I think that's very short. The process understanding, that is not that easy to reach. It needs first proper measurement. It needs consistent data, reliable data sets, information which are independent of scale and initial conditions, clever experimental designs to actually do a very structured process development work before actually we go to the next level of capturing the understanding of the upper part of the cycle in knowledge in digital twins, in models, and the models will be implemented in digital twins finally to achieve optimized predictive control and so on. So that is, let's say, the view. And of course, this is an old figure on quality by design. You see how many different disciplines and issues are actually involved. You see here the process design, product performance cycle. You see all the different specifications and how process parameters act on product specifications and quality attributes. All of this is linked to product knowledge and process understanding, what I just mentioned here on that. And of course, that never stops, and this is a continuous learning, and it's all embedded into continuous improvement. I think that is the main focus and is a methodological approach here. To also shortly introduce our EFCE Working Party on Quality by Design, you see here the major objectives which we try to fuel. We further promote the use of chemical engineering methods and tools in pharmaceutical process development operation. We try to offer a suitable forum for dissemination of research results. We try to coordinate research activities at the European level and identifying areas and problems that are important for development of novel and improved pharmaceutical production processes. We promote cross-disciplinary exchange of knowledge and ideas because we are very much, let's say, interdisciplinary here in this field. And of course, we actively see collaboration within other EFCE working parties and do, let's say, common activities here to above mentioned activities in practice. And we also, of course, what we do in practice here, we try to propose workflows, how to actually get QBD into practice. I just mentioned here a couple of these elements. We try to publish certain articles in this way, or to see how we actually get, let's say, from a methodological element like QBD is, to actually finally something which is really transferable to industry, because I think finally we are always just interested in transferable methods which really fly in industrial application. With this one, I want to introduce the first speaker here, which we have, and this is Pierantonio Facco of the University of Padova. And I think he's well known in this space in digital twins and machine learning. And with this one, I just stopped already with my short introduction, and would like to hand over to Pierantonio to start his talk. Pierantonio, the floor is yours.
Thank you very much for your nice introduction. I'm going to share my screen. Okay.
Yeah, perfect. Go ahead.
Okay. So this presentation deals with the application of machine learning techniques to aid the scale up in the biopharmaceutical processes. After a brief introduction on the biopharmaceutical industry issues, I will show how some machine learning techniques can be utilized for the development of a biopharmaceutical process, in particular for the production of immunoglobulins. At the end, I will conclude with the final remarks and some future work. The pharmaceutical industry is living a critical period. Typically, the drug development is really cost intensive and time consuming due to the highly regulated healthcare system. Now, with the spending review of the governments and the expiration of patents, the erosion of the market by generic, this is even a more difficult, a more challenging issue. However, there is a sector, which is the biopharmaceutical production, which is increasing its importance. Think to vaccines today, but it is not limited to vaccines. There are a lot of classes in which we can find interesting products such as blood components, tissues, recombinant proteins, and so on. The biopharma sector is increasing its importance. I think that they are increasing their revenues with very important annual sales, and that currently more than 7,000 drugs are on development. The challenges in the development of a biopharmaceutical product are related to the strict regulatory framework. So we have to, let's say, build the quality of the product within the product since its conception and the first stages of the development. So we wanted to reduce the time to market of a new product, launching into the market the new drugs in a cost-effective manner, and guaranteeing a scalable manufacturing process with high flexibility. And so we have to understand very well all the parts of our processes, and for this reason, modeling can be adopted, both first principle models or database models. The development in a biopharmaceutical industry is even more complicated if we think to the complex series of interconnected chemical reactions and biological phenomena. Furthermore, we are dealing with living materials, so we have to face high variability. Typically, the experimentation, an extended experimentation, cannot be carried out in the industrial scale. So the experimentation is very, let's say, difficult and slow because we have to scale down the process, and then in the lab scale we can optimize the parameters in such a way as to maximize the production, and then there is a slow scale up toward dozens of passages, and all these passages are critical to return to the commercial scale production plant. Today, I will show you one example in the development of a process for the production of immunoglobulin. We begin, the experimentation begins with high throughput experiments. They typically are held in the microscopic scales, where a lot of parallel experiments can be carried out with a relatively low complexity. Then, when we pass to the centiliters and deciliter scale, the complexity increases and the number of experiments unfortunately decreases. And when we arrive to the liter scale, very few experiments can be performed because of the complexity, for example, the complexity of the fluid dynamics, and the fact that automation is required, for example, for the control of pH, temperature, feeding, and so on. However, from all these stages, a large number of available data are present. In this case, we will see data coming from different scales, such as static scales, the microwave plates, some shaken scales, such as shake flask, or some stirred environments such as amber 15 or amber 250 and 2 liters bioreactor. From all of these stages, typically the measurements which are available are related to chemical or physical indices, temperature, pH, dissolved oxygen, a lot of biological indices, which are mainly related to the monitoring of the concentration of the cells, such as the cell titer, the cell productivity, or the vitality, let's say, of the cell. So, for example, the variable cell count, the viability, and so on. We could have also some data from omics, okay, and in particular, this data come from the experimentation made on different cell lines, different clones. So we perform one experiment per clone, and we assess information on all these variables at the end of the batch, or in a different time instant throughout the entire length of the batch. So in this case, we consider all the dynamics of the experiment. And the wealth of information of this data can be utilized to accelerate the process scale up and to aid the process development. For example, we can obtain an in-depth process understanding to understand what are the major driving forces and the highest sources of variability. We can select the most promising cell lines, which are the most productive and stable. And finally, we could also study the similarities and differences among different scales. Typically, so we exploit the data through machine learning with different type of activities. Some activities are related to the descriptive analytics to understand what happens in our biopharmaceutical process. If we want to increase the business value, we could adopt some diagnostic analytics to understand why those phenomena happen within our process. But if we want the highest business value, we have to increase the complexity of our methodologies and refer to predictive analytics and prescriptive analytics. So we don't, not only we wanted to forecast what will happen in our system, but we want also to understand how we can make something we desire happen. For example, how could we manage the critical process parameter in our system in such a way as to guarantee to obtain a quality of a predetermined target profile? Well, so all these activities, we organized all these activities in a framework that was recently published in the Biochemical Engineering Journal. And these activities are oriented to understand the relation between different experiments and how clones, cell lines perform, and how variables are correlated both within a single scale and between different scales. And in the next few minutes, I will show you a couple of examples of these activities. They are mainly related to the use of database methodologies, so pattern recognition techniques. But in this case, I will show you only the application of two multivariate statistical techniques, which are PCA, principal component analysis, or PLS, the projection on latent structure. I don't want to give you too much details on the mathematics. I can simply say that these are appropriate tools to deal with large multidimensional data where variables are highly correlated. And the rationale behind these techniques is that the data are projected in a low dimensional space made of latent variables, which are some hidden information that are related to the few chemical, physical, and biological phenomena that occur into the system. And in particular, I will refer to two mathematical entities, which are the scores and the loadings, where the scores define the behavior of the cell lines, while the loading retain information on how variables are correlated. The first example I would like to show you is the descriptive and diagnostic analytics to track the cell line performance in a smart manner. We apply the multi-way PCA on the amber 15 scale, and here we try to understand the dynamic behavior of the variables and the variables correlation through loadings for the entire duration of a batch. Here, a batch is approximately 2 weeks, here we monitor the variables in 8 different time instants, and we can see the relation between variables through the loadings. In fact, for example, we can see that the titer, this dotted magenta line, is highly related to the variables identifying the productivity and in the first part of the batch to the viability. This means that these variables vary. They vary together. When one increases, the other ones increase, and vice versa. Some other variables are entirely related. For example, you can see this dashed dot line indicating the glucose as a trend, which is the opposite of this dashed line, which is lactate. In this case, this means that when we feed glucose and the glucose is available in the culture, there is a certain well-being of the cell lines, and the lactate is low. While when the glucose is scarce, there is a suffering of the cell lines, and the lactate increases. Furthermore, the same modeling strategy can aid us to monitor the performance of different clones. For example, in the score space of the first 2 principal components. Here, 1 point indicates the relation between all the variables for the entire duration of an experimental batch. And so 1 point summarizes the information we have in one experiment. And here you can see that the cell lines, which are in red, which display an high IVC, can be clearly distinguished from the cell lines that in the experiment display a low IVC. In terms of titer, we can do something similar. So the cell line which are highlighted in green here are those which show an high titer, and they are very distinguished very well from the cell lines, those highlighted in blue, which display a low titer. But we don't want to think to the data one at a time in a univariate fashion. We want to map the performance of the clones with a joint and multivariate reading of all the variables at the same time. So here in the score space, we identify the zone of the cell lines with high performance. The cell lines here are highlighted with the red points. Here we can see all the cell lines that have high titer and high IVC, for example, at the same time, while the cell lines in black, indicated by black squares, are all the cell lines with low levels of productivity. And this mapping tool can be utilized to monitor the clone's performance in real time. In fact, if we project the experimental batch of one new cell line in this score space, here we can see the time point 1, time point 2, and how the experiment progresses until time point 8, which is the end of the batch. You can see that this cell line goes to the zone in which the low-performing cell lines are located. So this means that this cell line should be discarded, while in this new map, we can see that we projected the batch measurement for a cell line, which progresses to the zone, towards the zone of the high performance cell lines, indicating that this is a cell line that should be progressed in the scale up. Furthermore, we have another indication in this map, which is that while in the first 4 or 5 time points there is a progression of the status of the cells, in the last time points the status of the cell remains steady, meaning that the clone's history is already written after approximately 50–60% of the total duration of the experiment. So in 1 week, And this is the motive why we could cut the experiment and save time and resources. The second activity I would like to show you is related to a predictive analysis, and in particular to the prediction of the productivity performance of the cell lines in the scale-up. And to this purpose, we adopted a multivariate regression technique, which is the projection on latent structure. This is a scale-specific model in the sense that we could exploit the information we have on some variables which are easy to measure to predict the performance or productivity of the cell lines and the variables which are more costly or more difficult to measure. However, there is an improved version of this PLS modeling, the joint Y PLS, which can exploit not only the data available in one scale, but all the data available in the other scales of the scale-up process. And now I'm showing to you the scale-up from amber 15 and shake flask to a 2-liter bioreactor. And here you can see that well, sometimes the scale-specific model built with the PLS is good because it is scale-specific. So the prediction, for example, of the IVC in red points are always well close to the real measurement of the IVC. However, there are other indicators, such as the peak viable cell count, in which the joint Y PLS, so the model exploiting all the information available in all the scales, outperforms the scale-specific method, meaning that valuable information can be found in the other scales. And this is always true when we have only few data, few experiments in one scale. Well, so in conclusion, I can say that we developed machine learning techniques for assessing information about the most important driving forces within the biopharmaceutical process in all the scales. We developed tools for mapping the cell line performance in terms of productivity and identifying for the early identification of the most promising cell line to be progressed in the scale-up, and for the prediction of the productivity of the cell line also across scales. I didn't show it, but we also developed tools, model-based tools, for understanding if similar biological, chemical, and physical phenomena occur in different scales or not, and if the behavior of the clones remain stable across scales. But what's next? Well, typically, we have in all the stages of the development, we have information which are related to the engineering part, so the process development, the process scale up, the plant management, and information which are related to the biological world. These are intended to understand the biological features. We are intended to optimize our culture, to do some host engineering. Now, the next step is to find a link between the process and the biological information. So, for example, fusing the process measurement and some biological measurements, such as, for example, metabolomic data, to have improved methodologies, to develop improved methodology for the selection of the cell lines. So in this case, for example, we are working on the fusion of data on the amber 15 scale from the process and from the dynamic time profile of the metabolomics derived from spectroscopy. So we exploited this large amount of data not only to understand the relation between the process and the biology, but also to have an in-depth understanding of the metabolomic pathways. So we could link how the production of the specific immunoglobulin is related to some phenomena which are related to the energy production and the presence of some cofactors and vitamins, or the activity of some amino acids. But if we reverse our way of thinking, we could also optimize the process. So, for example, finding the optimal strategies for feeding, or finding the optimal process parameters in such a way as to deal with cell lines with specific metabolic features, or even try to modify the cell to be more robust to the process variability. Well, I have concluded. I would like to thank you for attending this presentation. I would like to thank also GlaxoSmithKline, and in particular the Biopharm Process Research, for letting me share with you their case study. And I would like to thank also these two guys, Gianmarco and Francesco, who helped me with this project. Thank you very much.
Thank you so much, Antonio, for this nice and dynamic talk. Very nice. Thank you so much. I wonder if we put the questions now or later. I think it would be probably good to have one question, if you allow, and this came in here from Jaka. Thanks for being there, Jaka. I don't know if you see this question. It's mainly, let's say, when you use data from different scales, what confidence do you have in the accuracy of the prediction at scales when you have actually only a few examples or few data available for calibration of your models across the scales?
Okay. Well, I presented a well-established regression technique, which is intended to the transfer of different products, from one scale to the other, or from one side to the other. This improved PLS modeling, which is joint wide PLS, could exploit the information we have in the other scale, finding the correlation between different variables in one scale and their correlation also with the other scale. So this correlation is typically well established. Also when you have few data. And furthermore, you could try also to characterize the uncertainty you have. And well, there are some methodologies to characterize the uncertainty and take into account of the uncertainty.
Mm-hmm. We will probably come back to also the other points, Jaka, which you mentioned. I think there's overarching points also, how to test on independent test batches. Let's say, how do you test that? How do you validate your approach, etc. I think these points are overarching elements, of course, of all kind of models and so on. I think would like to take 5 minutes at the end that we can probably discuss all these points together. You allow, Jaka, also. Thank you very much for that talk already. And that would be, I think, a very good start to hand over